In this video, I'll discuss sections 3.3 and 3.4. The learning objective is to learn about methods to characterize and sequence peptides and proteins. Polypeptides contain differing amino acid sequences, and the sequence and arrangement of those amino acid residues gives the polypeptide a chemical character. And some polypeptides bind specific targets, which can be used to fish them out of a complex mixture. A mixture of proteins can be separated on the basis of differences in physical and chemical properties. For example, differences in charge, size, affinity for a ligand, solubility, hydrophobicity, and thermal stability. Chromatography is commonly used for preparative separation, in which the protein is often able to remain fully folded. This illustrates column chromatography. This allows the separation of a mixture of proteins over a solid phase using a liquid phase to move the proteins through the solid phase. Proteins that have a high affinity for the solid phase will remain on it longer, and they will be removed from the column later. Those with a lower affinity will wash off first. Typically, what happens is that the protein solution to be separated is layered on top of the column and allowed to percolate into the solid matrix. Then additional solution is added on top. The protein solution forms a band within the mobile phase that is initially the depth of the protein solution applied to the column. Then as the proteins migrate through the column, they proceed at different rates based on their different interactions with the solid matrix material. In this picture, protein C is washing off of the column first, which means it must have had the least affinity for the column. And then that will be followed by B, and then finally followed by A. So A must have had the greatest affinity for the column because it took the longest to be released from it. Another type of method for separating proteins is to separate them by their charges, also called ion exchange. In this method, you have a column that contains polymer beads with, if it's a cation exchange column, negatively charged functional groups. So there are cation exchangers and anion exchangers. So with the cation exchanger, the proteins that are positively charged will have an attraction to the negatively charged resin, and therefore they will move much slower through the column than the compounds that have no charge or have a negative charge. The compounds that have a negative charge will be repelled by the negatively charged resin and will move through faster. Also note that the magnitude of the charge affects the rate of flow through the column. So in this case, the larger the positive charge, that is going to move the slowest, versus something that also has a positive charge but not as much of one. And then also the largest net negative charge species will move through the fastest. Another type of separation method is size exclusion. And in this, the column contains a cross-linked polymer, and this is a picture of what that might look like. And small proteins will get caught up in the cross-linked polymer and move through the column more slowly, whereas larger molecules will pass more freely. So this method separates proteins by size. Another method for separation is the affinity method, affinity chromatography. And in this, the beads in the column have a covalently attached chemical group called a ligand. And the ligand is simply a group or molecule that binds to a protein. And when a protein mixture is added to the column, any protein with affinity for this ligand binds to the beads, and its migration through the column is slowed down. On the analytical scale, separation is often done by electrophoresis. In this, an electric field pulls proteins according to their charge, and the gel matrix hinders the movement of proteins according to their size and shape. The most common gel used is called polyacrylamide, and separation of proteins via electrophoresis is often called polyacrylamide gel electrophoresis, or PAGE. Electrophoresis as an analytical method is extremely important, and its advantage is that proteins can be visualized as well as separated, allowing a researcher to quickly estimate the number of different proteins in a mixture or the degree of purity of a protein preparation. This is a picture of what it would look like. Different samples are loaded into the wells or depressions at the top of the polyacrylamide gel, and then the proteins move into the gel when an electric field is applied. And typically the pH is adjusted so that the proteins will have a negative charge and move in the direction of the positively charged electrode. In one form of polyacrylamide gel electrophoresis, the detergent sodium dodecyl sulfate, shown here, is added to the protein solution. It will denature proteins, and the proteins will unfold. Proteins assume a rod-like shape in the presence of SDS, and most proteins bind SDS in a ratio of about 1.4 grams per gram of protein. Notice that SDS has a negative charge. The large negative charge that the SDS imparts will end up masking the protein's intrinsic charge, and the net result is that SDS-treated proteins have similar shapes and charge-to-mass ratios. Therefore, the separation is going to be based purely on the mass. Therefore, this method is particularly useful in determining the mass of proteins. And smaller proteins will move faster, larger proteins will move slower. This is an example of what a particular gel might look like after being stained blue with a stain that adheres only to the protein and not to the gel itself. Each of these areas here we call lanes. And this particular example refers to the purification of the Rec-A protein of E. coli. In this, the gene for Rec-A protein was cloned so that its expression, its synthesis of the protein, could be controlled. The first lane shows a set of standard proteins of known mass, and those are going to be our markers. The next two lanes show proteins from E. coli before and after the synthesis of Rec-A protein was induced. The fourth lane shows the proteins in crude extract, and the subsequent lanes show the proteins after successive purification steps. So the electrophoretic mobility of a protein on an SDS polyacrylamide gel is related to its molecular weight. So here we see a gel where the proteins have been stained in blue. So you will treat the gel with a stain that adheres to the proteins but not the gel itself. And in lane 1, we have standard proteins of known molecular weight. And then these marker proteins are guides that help us to estimate the molecular weight of an unknown protein, shown in lane 2. So these areas here we call lanes. And a plot of the log of the MR of the marker proteins versus the relative migration during electrophoresis is linear, shown here. So you can make that plot and then figure out the molecular weight of the unknown protein. Isoelectric focusing is a technique that separates proteins according to their isoelectric points. So in this, a protein sample is placed on a gel strip containing an immobilized pH gradient. So here you would have pH, lower pH on this side, higher pH on that side. With an applied electric field, proteins enter the gel and then they migrate until they reach a pH equivalent to their pI. Because remember, when the pH equals pI, the net charge of a protein is 0. Then once you stain it, you can see the proteins, and they're going to be distributed along the pH gradient according to their pI values. Another technique is two-dimensional electrophoresis, where we're combining SDS page and the isoelectric focusing. So with this, the proteins are first separated using the isoelectric focusing in a thin strip gel. The gel is then laid horizontally on a second slab-shaped gel, and the proteins are separated by SDS polyacrylamide electrophoresis. Horizontal separation along here represents differences in the pI, and vertical separation represents differences in the molecular weight. Aromatic amino acids absorb light in the UV region, and proteins typically have UV absorbance maxima around 275 to 280 nanometers. Tryptophan and tyrosine are the strongest chromophores. If you look at their structure, you see that they contain aromatic rings. For proteins and peptides with known extinction coefficients or sequences, the concentration can be determined by UV vis spectrophotometry using the Lambert-Beer law. This shows a comparison of the light absorption spectra of the aromatic amino acids tryptophan and tyrosine at pH 6. The amino acids are present in equimolar amounts under identical conditions. Notice that the measured absorbance of tryptophan is as much as 4 times that of tyrosine. Light absorption by the third aromatic amino acid phenylalanine generally contributes little to the spectroscopic properties of proteins. Proteins in a complex mixture often require more than one purification to completely isolate the protein of interest. If a protein has a specific function, such as binding insulin, the fraction that binds insulin best after each purification step will contain the most of the protein of interest, and the function of the protein is called the activity. The ratio of the activity to the total protein concentration is called the specific activity. The difference between these terms, activity and specific activity, can be illustrated by considering 2 flasks containing marbles. The flasks contain the same number of red marbles, but different numbers of marbles of other colors. If the marbles represent proteins, both flasks contain the same activity of the protein represented by the red marbles. The second flask, however, has a higher specific activity because the red marbles represent a higher fraction of the total. It is very important to know the sequence of amino acids that make up a protein. Now, in modern times, the actual sequence is generally determined from the DNA sequence. There are other methods that you should know about. The classical method is called the Edman degradation, which involves successive rounds of N-terminal modification, cleavage, and identification. And it can be used to identify a protein with a known sequence. The other method is mass spectrometry, which is a modern method that is commonly used. And this can precisely identify the mass of a peptide and thus the amino acid sequence. It can also be used to determine post-translational modifications. In the Edman degradation, phenyl isothiocyanate, this molecule, reacts with the N-terminus, the amino terminus of a protein or polypeptide, and it cleaves that last terminal amino acid from the rest of the polypeptide. In doing so, it forms this derivative where R1 corresponds to the side chain of the amino acid nearest the N-terminus. Then that is further treated with acid and identified. So based on what this is, you can figure out what the amino acid at the N-terminus was. And then you essentially do this over and over and over again, reacting the amino acid at the N-terminus with phenyl isothiocyanate, cleaving that last amino acid, analyzing it, and then repeating. Now, in sequencing larger proteins, it's important to break them down into smaller peptides. And this can be done using a variety of proteases with specific cleavage points. And this is a table of example reagents that could be used and shows where they would be cleaved. Mass spectrometry has emerged as the dominant technique for characterizing and sequencing proteins, and it accurately measures the mass-to-charge ratio for ions in the gas phase. Until about 1985, macromolecules such as proteins could not be analyzed this way because the heat that was required to enter them into the gas phase was so high it would actually destroy the protein. However, newer techniques have addressed these shortcomings. For example, electrospray ionization, shown here, involves using a solution of a macromolecule such as a peptide, and it's sprayed from a narrow capillary tube maintained at a high voltage, which forms fine, highly charged droplets from which the solvent rapidly evaporates. This yields a series of gas phase macromolecular ions that typically have ionic charges in the range of +5 to +2. The charges result from the protonation of basic side chains such as arginine and lysine. The ions are directed into the mass spectrometer, which measures the mass to charge values with an accuracy of about 0.01%. Then consequently, determining an ion's charge permits its molecular mass to be determined with great accuracy. This is an example of what the chromatogram would look like, where you have the relative intensity on the y-axis and the mass to charge value on the x-axis, and you have a series of peaks. Another technique is tandem mass spectrometry. In this, you start with the electrospray ionization, which generates gas phase peptide ions. And then these are separated by the first mass spectrometer. Then one of them will be directed into a collision cell where it collides with helium atoms. And this treatment will break the peptide into a variety of fragments, which are then directed into a second mass spectrometer to determine the mass to charge values of all of those fragments. Sequences of proteins with identical functions from a wide range of species can be aligned and analyzed for differences, and such differences can indicate evolutionary divergences. An analysis of multiple protein families can indicate evolutionary relationships between organisms and ultimately the history of life on Earth. In summary, in Chapter 3, we learned about the many biological functions of peptides and proteins, the structures and names of amino acids found in proteins, the ionization properties of amino acids and peptides, and methods for the separation and analysis of proteins.